Hi, my name is Carlos Vegas. Thank you for taking the time out to come to my presentation and learn about my research and the research we do here in Dr. Albajani's lab. I'm a neuroscience major, and my research is mainly focused on nervous system regeneration, specifically elucidating the mechanisms in which RAP 6.2 inhibits axon regeneration in C. elegans. That's a mouthful, so let's break that down. So first, I want to talk about what is axon regeneration and why is it important? Well, axons are the long tubular structure that neurons use to communicate with one another. And when these axons are injured, neurons are unable to properly communicate with one another if they don't regenerate. And unfortunately, our central nervous system, which make up our brain and spinal cord, experience little to no regenerative capacity. Thus, when there's injuries to these areas, specifically through injuries such as traumatic brain injury and spinal cord injury, these structures, these axons are damaged and are unable to regenerate and reconnect and communicate with one another. And when this happens on a large scale, it causes physical and behavioral dysfunction that we see in individuals with traumatic brain injury and spinal cord injury. And my research mainly focuses on finding the factors that either promote or inhibit axon regeneration, either inside or outside of the neuron. And I'm doing this in the model organism C. elegans. C. elegans are small, 1 millimeter long, transparent worms. We can easily genetically manipulate them, adding genes, removing genes, and we can easily visualize axons and cut them because of their transparent skin. And they also reproduce very fast, which is very convenient for doing a lot of experiments. Right here is a little image of how we cut axons. So we use a pulse laser system in which we visualize these animals under a very high magnification microscope and sever axons in the middle of the worm or the midline of the worm. And following 24 hours, I score regeneration as either positive or failed regeneration. And I do this by looking at for signs of regeneration. And mainly the two signs that I look at are if the axons have grown past the area that I've cut or past the midline, and if they exhibit a structure called a growth cone. And this is a web-like protrusion that looks sort of like a hand sort of reaching out to try to reconnect. And axons that have these structures, I score as positive, and those that don't, I score as failed or no regeneration. So what is RAP 6.2? RAP 6.2 is a gene that encodes for a small protein found within the cell, primarily within the Golgi of the cell. And it mainly functions to move around different proteins and enzymes around the cell. And we know that this gene inhibits axon regeneration, but how it does so, we don't fully know. It's also highly conserved with its mammalian counterpart, RAP6A, indicating that figuring out how this gene works in C. elegans may have implications to mammalian systems, such as humans. So how does RAP 6.2 inhibit axon regeneration? Currently, we only know one possible mechanism or one mechanism in which it functions through, and that is through a protein called APL1, and it does this within the neuron. But my research is mainly focused on what's happening outside the neuron, specifically the skin, the epidermis layer of the skin. So why do I care about the skin? Or why do we care about the skin? Well, a previous paper in our lab showed that RAP 6.2 actually works within the skin to promote its integrity and strength. And when you remove this gene, the animals are much more fragile. As you can see here in this image with the animals labeled RAP 6.2, when you put them in water, they rupture much more easily due to this compromised integrity of their skin. And when you express this gene in the skin specifically, nowhere else, it recovers this fragility. They get back their integrity and strength of the skin. So one of the first questions I asked was, is fragility responsible for the inhibitory effects of RAP 6.2? And the way I went about answering this question is by testing regeneration of a gene called BUS17, which exhibits identical skin fragility issues when mutated. And I found that BUS17 mutants did not have different regeneration levels to wild type or animals with a functional copy of BUS17, indicating that fragility is not responsible for the inhibitory effects of RAB 6.2. Next, I looked and asked the question, does RAB 6.2 function in the epidermis to inhibit axon regeneration? So what I did was I expressed RAB 6.2 specifically within the skin, or the epidermis layer of the skin, and nowhere else, and compared the regeneration of those animals to animals without RAB 6.2 and animals with a functional copy of RAB 6.2 or wild type. And what I found was when I expressed this gene only in the skin, I found a reduction in regeneration. But this reduction in regeneration was not significantly different from animals with RAB 6.2 and animals without RAB 6.2, giving me inconclusive data. And I think that this is mainly because of the sensitivity in which I scored regeneration wasn't sensitive enough to tease out this difference. As I said before, I scored regeneration as either positive or failed regeneration. So it didn't account for the extent of regeneration or how much the axon truly regenerated. And also I have two graphs here because I replicated this with two different strains of animals. And so a future study I would like to do is actually quantify the length in which these axons regenerate, measuring it from the bottom all the way to the tip of the regenerating axon, and then factoring in the diameter of the worms. And I think this will help me answer my question. And thank you so much. That's the end. It was a very short, brief overview of my research. And if you have any questions about it, or you're interested in learning more or getting involved, please email me or email Dr. Albajani. Thank you so much. Thank you for coming again. Bye.